Hello, I'm Ushma Neal with the Journal of Clinical Investigation series, Conversations with Giants in Medicine. I'm happy to welcome today the Johns Hopkins-based physician scientist, Dr. Elizabeth Jaffe. An oncologist by training and an immunologist by trade, Jaffe led the translational development of a first-gen cancer vaccine for pancreatic cancer and continues to innovate in the cancer vaccine space. She embraces the convergence of new technologies and approaches to uncover the complex signaling pathway between tumor cells, their microenvironment, and the immune system towards the development of novel cancer immunotherapies. All the while, she has acted as a leader at the national and international level, currently serving as the chair of President Biden's cancer panel. Thank you so much for joining me today, Dr. Jaffe.
Thank you so much for inviting me. I'm honored.
Well, why don't we start with the beginning? Can you tell us a little bit about where you grew up and about your parents and your family?
Yes, I have humble beginnings, which I'm very proud of, I think, because I think many of us in America, started from humble beginnings. Both my parents were children born during the Depression. So both families were poor. My father was from an immigrant family from Russia. And my mom's family had been here for a generation, but still the Depression hit everyone. I was born in Brooklyn. And at that time, my mother and father met when my mother was 19, my father was 22. My father had worked his way through college, lived at home, went to Baruch Business School, which is City College. And my mom did 1 year of college, but that was it. So to be honest with you, I'm the first woman in my family to have gone to college of all my female relatives. But my dad had trained as an accountant. My mother was a homemaker. And my dad then was drafted into the Army. When he came back, they had me. And then he decided to start his own business. I had an uncle. My great uncle, his uncle, who was like a father to him and gave him a very small investment to buy a house in a poor neighborhood in the Bronx, and he rehabbed it and rented it out. And he was starting his own business in real estate, essentially, was doing well. And I'm the oldest of five. They had three kids. We moved from a one-bedroom home to a three-bedroom, two-family home, which was very small. But that was a huge advance for them. And that was still in Brooklyn, in Canarsie. And my father got shot while collecting rent in this building in the Bronx. So he was laid up for a year. In those days, I guess surgical techniques were not very good. It was the early 60s. And it was mid 60s. I was in first grade. It was traumatizing. My dad was pretty ill for quite a while. It seemed like almost a year. My mom had to go back to work. They now had four children. So it was very stressful. Grandparents were trying to help out, but my grandfather was pretty ill, one of my grandfathers. So there was that complication as well. Very close family, lots of people in Brooklyn and Queens and the Bronx, family members. So still, I had a very loving environment. And then, my dad got better. His business took off again. He had a nice partner who was maintaining it at the time. And then they had my sister, a fifth child. And we moved to Long Island, where we could use public schools. I mean, my father wasn't extremely wealthy that he was going to send us to private schools, but we went to Long Island, had good public schools. And at that point, I was in middle school. So, being on Long Island, not being able to walk anywhere, I had to make new friends. Actually wasn't as much fun as Brooklyn, New York was for me. I think my brothers and sisters were fine. I made friends, but they lived far away. My mother had five kids, so to drive me places. So it was an adjustment. But then high school came around, and I think I put a lot of my efforts into, I love to read, and I put a lot of efforts into, just schoolwork and school activities. So staying after school for different clubs and stuff. But nothing that as a hobby long term for me from that time in my life. But the one light in my life was that the second half of senior year, I met my soon to be husband 4 years later. So we were high school sweethearts, and we went to the prom together. And it turned out, we both ended up in Boston for college. And so we kept together. And so I think of all the things that were not so great in high school, that was certainly a bright light for me.
Wonderful. So along this trajectory, I know you landed at Brandeis for college. How did you decide what to major in? And were there any roots of your scientific and medical interests at that point?
I've been interested in science since third grade, when a great uncle developed cancer. And I remember my grandmother and grandfather, who I was very close with, were so upset. His name was Happy, by the way. I just couldn't imagine somebody with the name of Happy, of course it was his nickname, could die of cancer. So, third grade. And that's another piece of trauma in my life that I remember. And so, as a young girl, it was hard to find mentors and to go into science. But I had a fourth grade teacher who she was a math teacher, and she noticed that I was very good in math. And also she would talk a lot in class about, the race to land on the moon. This was, mid 60s. So, science was very prominent in schools then. I mean, people were starting to think, oh, science is a good field. And that's when I read Marie Curie's biography, and I'm thinking, wow, this woman not only was a woman in those days who identified something new, but it turned into a field in cancer. I mean, really, she's the mother of radiation oncology. To me, that was just, it was like a bright light again went off in my head. Wow, can I even do something similar? So I didn't have any people who were alive then who were my mentors, but she was basically my mentor in a sense early on. And then I meet this fourth grade teacher, and she's encouraging me to go into math or science. So I think that was enough. There was a person paying attention to me and, my science. My mother didn't understand any of this. My dad was focused on taking care of 5 kids, and he took care of lots of family members as well, very poor family, due to people coming out of the Depression and not really having jobs. So my father was always taking care of people, and which was wonderful. And that's something I learned from him, and probably maybe why I went into a field where you take care of people. But, it wasn't like my parents encouraged me. Not that they discouraged, they just didn't encourage me. And then I think what happened was I met yet again another teacher in 11th grade. She was my chemistry teacher, again, a woman, chemistry. She loved it, the passion that came out from her. And again, there were very few girls in the class. I mean, we were still girls, but she encouraged me. And that was really wonderful. And so I had that. And so I thought, well, chemistry was a lot of fun. I love chemistry, but I really wanted to go into the biological sciences. So what ended up being my major, and I chose Brandeis because it was one of the top biochemistry undergrad majors in the country. It was a small school. My high school, we had 900 graduating. Way too many. I was such a shy person. So, we had class sizes sometimes of 40 kids. And so unless a teacher got to know me, I wasn't one of those kids who were constantly talking in class. And so a lot of teachers didn't appreciate who I was or what I could do unless they got to know me. But, going to Brandeis, I started out in science classes and other classes where there were, 20 to 30 people in the class. So even freshman year, I had a introductory biochemistry professor who invited us over to his house. It was really a perfect place for me. So I got in, of course, early decision, thank goodness. And so I decided I was going there, and I wanted to be in Boston. And it was probably as far as my parents could conceive of me leaving, because they had no context of kids leaving the house and going away to college. So it worked out really well. And my husband ended up, finding out a bit later where he was going. So he ended up in Boston as well. And so we were able to keep in contact. So it was a fun experience in college. At that time, There were some amazing, there still are amazing scientists, but I was able to identify a young assistant professor who was doing immunology, and I wanted to learn immunology. So I guess it was my second year. I went to her lab, and this was 1979. So a couple of years earlier, a very major advance was made, and that was the major advance. Milstein and Kohler identified hybridoma technology. So remember, T cells at that time we weren't even thinking about. These were B cells, and how are B cells turned into antibody producing cells. And so we had come up with a project where I would develop hybridomas from immunized murine B cells and then fuse them and make hybridomas and hopefully screen and identify a heavy chain switched antibody, which of course we found a number of them. And the concept was, could we study heavy chain switching with this technology? And the answer was no, we needed genetic technology to really do that. And that happened about 10 years later, but it was a great learning experience. And, I enjoyed the lab. It was a tremendous experience. And the one other college experience that I had was that David Baltimore, who won the Nobel in 1975, I think, on virology, he came and gave a talk to one of my classes at the time when I was a sophomore as well. And he talked about virology, and I was so excited. And it was at that point that I realized, I'd always thought that the immune system was powerful. I'd always thought that the immune system was one of the most important systems for even therapeutics, because when you think about the two greatest biomedical achievements in the 20th century, one was penicillin, right, antibiotics, and then the second was vaccines that prevent kids from dying in childhood. So, we knew vaccines activated the immune system. Did we know how? Not necessarily, but he started talking about that. So I started putting two and two together and thinking, well, if the immune system can see a virally infected cell, why can't the immune system see a cancer that looks different from the normal cell? So that sort of got me started on my pathway toward immunotherapy and cancer.
At this point, though, you're steeped in research, you're having all of these ideas. So what drove you to go to medical school, or was that just the natural next step for most people at the time?
I'd done some volunteer work as well, working with kids with cancer. And I liked the clinical aspects of that work. And so I wanted to really experience what it's like to be a physician. And so I was, medical school seemed to be the right place for me. My husband and I decided we would get married. It was 1981. You don't exactly live together in those days, at least not with an Italian mother-in-law to be and a Jewish mother-in-law to be. So it was one of those situations. We were in love. It didn't matter. And, he has passed, but he was my best friend until that point as well. So anyway, so we went to New York because there was the most medical schools there. And, that way we would both have a good chance. I went to New York Medical College. He went to Columbia. We lived in New York. And it was a marvelous experience. I was able to train at two wonderful hospitals, one in Greenwich Village that took care of patients in all different areas, including Chinatown, elderly people. We went to prisons. It was quite an experience. And the other was in Harlem at Metropolitan Hospital, where you saw, as well, a different aspect of healthcare and what it's like to be a poor person and not have the best access. But this was a good hospital. It's still there doing, I think, good work for the community. So it was a marvelous experience for me to be in New York. The other very important experience, of course, was I was in medical school from 1981 to 1985 in New York City. And so we saw the start of the AIDS epidemic. And I learned a lot. It was sad. It was depressing to see young, mostly young men coming in and dying within weeks or months, or people who were IV drug abusers. It was one or the other. The hospitals were overwhelmed. Nobody understood the disease initially, so I watched it unfold. I watched it go from being called gay bowels disease, from being an immunocompromising disease where they were getting these diseases like pneumocystis, to an AIDS, to a virus that was identified. It was AIDS, and we even understood a little bit about how this virus infected the immune system. And then, within a few years, we saw how quickly drugs were developed against this virus.
Okay. Yeah. So talk a little bit about the move to Pittsburgh and your time there, because then near the end of your residency, there was also the year that you spent essentially doing research. So that time must have been equally impactful on you in terms of your career trajectory.
No, it definitely was. And so we were in New York for 4 years of medical school. All we saw was patients with AIDS. So we wanted to be internists, both of us. And so we decided we had to leave New York, which I think was wonderful to get experience elsewhere. And so it turned out that University of Pittsburgh at the time had a chairman of medicine, Jerry Levy, whose wife was also a physician, and there was no couples match at that time. And so he offered to give us our 2 weeks vacation every year, the same, which many programs were not willing to commit to. It's hard. It's hard because we were every third or every fourth night at that time. And so, and also Jerry Levy had this potential 4th year that was an NIH funded, like a T32 almost, grant, where it was sort of like the first year of a PhD where you rotate through different labs and you take didactic courses and learn different skills. And then you also conduct a year-long research project. And that's when I met John Kirkwood. And John Kirkwood had just come to Pittsburgh while I probably was a junior resident when he came. And talk about a person who is passionate about cancer and immunotherapy. He was so passionate. I mean, he just got me really excited. And so I ended up doing a longitudinal project with him and one of his junior faculty, and really enjoyed that year. And after training for 3 years and realizing that we had no good treatments for cancer, remember this was the mid to late 1980s, there was nothing. I mean, Taxol didn't even exist. There was absolutely nothing. So I really thought, wow, I can't just treat patients. As much as I would love to treat patients, I think I would be so depressed all the time, not being able to offer them hope, right? Not being able to offer them something that gave them potential to live a longer life and also a higher quality life. That's the other thing. Chemotherapy, I realized from the start, this is not a good quality of life with most chemotherapies at the time. And so that led me to wanting to do a research fellowship. And John and others suggested I look at Hopkins. I looked, of course, at a number of places. And my husband and I both had to be accommodated. He was looking to do a master's in epidemiology, so he needed a good school of public health. And when I was a second-year fellow, I was looking for a reagent that I needed, a plasmid, and I'm looking in the literature. And who has all these reagents? Dan Nathans here at Hopkins. So again, I was still shy. So I called his assistant and I said, does he have a lab manager I could talk to? I didn't want to bother Dan Nathans. He's a Nobel Prize winner. Remember, we had no computers. 10 minutes later, I get a call. Dr. Jaffe, I'd like to hear about your project. Tell me about it. And then I could tell you what you may need because I have a number of these plasmids. It was like Dan Nathans. He just called me up. I tell that to people when they interview here because that has been my experience here my whole career. It didn't matter that I was just a fellow. I was treated as if I was another colleague who was doing research, and everyone wants to hear about your research. That's how I got into pancreatic cancer. I got into pancreatic cancer because I was doing genetically engineered tumor vaccines, and that was my first project, working, with a team here. But we were also working with a team up at MIT. So we had developed preclinically, and it was Drew Pardole here and a few other people. And myself, and we had developed retroviruses that were actually the retroviruses were developed by our colleagues at MIT, but we had developed the ability to transduce tumor cells to express these cytokines. And then I developed the human vaccine. And originally, I developed the human vaccine with Frey Marshall, who was the kidney cancer surgeon, the senior surgeon here at Hopkins at the time. And he was very excited about what we were doing. So he said, why don't we do this first in renal cancer? So we said, sure. And, both renal cancer and melanoma were two cancers that were thought to be immunogenic. So it made sense to start there. And so I would go to the operating room and this doesn't happen anymore. And he would say, okay, he would take the tumor, put it on the side, and he'd say, what piece do you need? Because I needed a lot of tumor cells to develop the human vaccine. And then he'd come after the OR and come to the lab and take a look, and we'd talk. And science was fun. This is fun. I wish we had more time to do this. I try to still do this with all my people, but, it's harder these days than it was then. But we were having a good time, very quickly developed the vaccine. We tested it in patients. We had some evidence of activity. We were able to study the vaccine. And then Frey leaves. And this is 1994 now, so I'm on faculty for maybe a year or two. And Bert Vogelstein comes to my office and says, we're putting in a GI spore. So the spores had just started. It was the first GI spore. And he says, we're trying to put people together to do this. We'd love for you to be one of the career development awards to develop a vaccine for pancreatic cancer. And John Cameron, the surgeon here who, had really perfected the surgery, he comes and he says, Liz, you have to do this because I've done everything I can for surgery, and now it's time to get some experimental therapies. So they put together a group that included Ralph Ruban, who was a young pathologist at the time. He was doing transplant pathology, and Scott Kern, who was just coming out of Bert's lab as a geneticist and pathologist. And so they would get us all together, and probably a few other people, a few other surgeons. There was no clinical research at that time in pancreatic cancer. There were a couple of leaders in the country. There was Margaret Tempero, there was Dan Van Hoff, Jim Abruzzi. That was it. And they were doing some clinical research, but there was nothing innovative really coming to the clinics on a regular basis. And so this was my opportunity. And my father's brother had died of pancreatic cancer when I was a resident. So I felt like, this was my calling now, pancreatic cancer. And so that's how I got into pancreatic cancer. It was great working with all these people. And, my career just after that really kept moving forward. It was very, very interesting.
So Liz, as you were talking about that, it sounds like it was this perfectly linear upward trajectory of success after success after success, but very obviously, it must have had some challenges along the way, including just even the acceptance of cancer vaccines as a potential therapy and, immuno-oncology kind of bursting onto the scene. So I asked this question partly because I think the trainees out there absolutely need to hear it was not that easy.
No. So early on, when I had developed the genetically engineered cancer vaccine, it was the gene therapy community that embraced us. And so I remember going to meetings where Carl June and I were the only ones talking about cancer with gene therapy, because there's a lot of gene therapy going on for genetic diseases. And Carl June was a young guy at the time, and he was talking about the precursor to CAR T cells. And I talk about the vaccines, and, I got to know him, which was a wonderful experience. We're good friends as well. But I always tell people in the 90s and early 2000s, yes, we had our immunology communities, and I would go to Keystone meetings and see all these wonderful people like Jim Allison and Steve Rosenberg and all those people, and I'd get to know them. But, at these bigger meetings like AACR or ASCO, I always tell people that, when we were all invited to speak at these meetings, we were usually the last session of the last day. Nobody showed up except the other immunologists, right? So it was really transformative when all of a sudden these immune checkpoints were, making their way to the clinics. I think, we all knew about them in mouse models, but I don't think, the clinical world had really embraced immunology. I would always hear, immunology will never, be a therapy for cancer patients. And we all heard that over and over again. And then all of a sudden, this breakthrough and Jim Allison presenting patients who were responding to anti-CTLA-4 at these meetings. And all of a sudden it went from night and day. All of a sudden immunotherapy was important again. For me, what it made me realize, because we also learned a lot about T cell biology. So what I was realizing was I could do these vaccines, and we were seeing T cell responses in our patients, particularly ones with minimal residual disease. And we identified antigens, and we showed T cell responses to those antigens in patients who did well. And I have a group of patients who are more than 10 years survivors of, pretty advanced pancreatic cancer. But those numbers were small, right? And they continue to be small, but they're happening. But what I didn't realize was that we needed these immune checkpoints to keep those T cells activated when they get into the tumor. And so for me, it was an aha moment, right? That all of a sudden, okay, maybe these vaccines will have a role, even though a lot of people are saying vaccines don't work. Maybe adoptive therapy will be the way. Steve Rosenberg had some good successes with his adoptive therapy.
It occurs to me over the course of hearing you say that, and reading over your CV, the 6 patents that you have, the entrepreneurial side. Can you talk a little bit about the balance of all of those things? Entrepreneurial, clinical, administrative, running a lab, mentorship, and maybe sleeping at the same time? And what are your strategies for doing all of that? Or is it just because you have so much enthusiasm for all of these things and they exploded all at the same time?
Yeah, balance is an interesting word. I think we do try to balance, but we also have to make choices and prioritize. And so as a woman coming up in this field, I wasn't a pioneer woman. I wasn't the only person in medical school class or residency, but still we were under 10% typically. And so we were not encouraged to have children. So my husband and I waited 16 years before we had our children. We were way into our assistant professorship careers at that time. So balancing lab and clinic at the time was something I could do, and I was starting to review for the NIH. And the first time I was asked to review for the NIH, it's usually an ad hoc, and I enjoyed it so much. And that told me that, that would be one of my areas that I would continue to want to pursue in addition to my own research and some of the work I was doing for the Cancer Center in the School of Medicine. But so having kids, it kind of, I thought I was having one, we were going to start with one, and I forgot we had a very strong family history, and, I was gifted twins, which was wonderful, but it certainly complicated my thought process on how I was going to do this. My husband and I had older parents at this point, so it wasn't like they were going to be able to help out. But of course, we were making more money, so we would be able to pay for help. But I had to start thinking. I had also gotten a series of grants at that point, and I had spent more time on the clinical research than I was on developing my laboratory. And, at that time, I'm going to be honest, I was pregnant, and it was hard to get trainees to want to work with me because I guess at that time, people assumed pregnant women took off many months and wouldn't be available and those sorts of things. But, soon after I started building my lab, I got an amazing oncology fellow came to my lab, Dan Lahiru, who said, you know what, I want to do pancreatic cancer with you. And he really liked the clinical research. So he became this partner in clinical research, and he allowed me to step back from the clinics. And we truly have been a partnership ever since. And, he's the clinical person. He's probably the clinical expert in the world at this point in pancreatic cancer. And I was able to maintain my interest in the clinical part of things through being able to hold the INDs for the clinical trials, help him develop the clinical trials, and then I, of course, analyze the patient specimens. So although I don't directly see patients anymore, and that's when I decided to step back because I had kids, I had an active lab, I started getting graduate students and some postdocs, and I had this wonderful person who wanted to be my partner in the clinics. And that's, I'd always believed in team science, but this started my whole belief that you can be a part of lots of things. And Dan felt the same way if you worked in teams. And the whole trick to that is giving everyone, the accolades, the credit.
So it then strikes me that you were a natural choice to be a leader because you want to get things done and you don't need the credit for it because it's more about getting to the end. But in reading over your CV, just seeing the sheer number of leadership roles you've taken on at Hopkins, at the NCI, as president of AACR, and then more recently working with both the Obama and Biden administrations. So I'd be really curious to hear a little bit about your leadership style, if you want to elaborate. But beyond that, what was it like meeting Presidents Obama and Biden? Because I think for many of us, like that's such a thrill and to have that kind of a platform. But then further on from that, like how it is you managed to synthesize so many different people's perspectives and points of views and opinions to come up with a set of priorities at any of these levels.
I love translational research. I love mentoring, but I also love working with other people and learning from them. So I'm always learning. And I've realized that I much prefer the translational research arena from the administrative. And so a lot of women have said, why aren't you cancer center director? And, I've had opportunities, but I think my leadership is to really facilitate other people, work with other people, learn from them, and help, as you point out, synthesize new innovative concepts. And I just love when I can develop a new program, when I could be a part of the Blue Ribbon panel. And again, that it just, being in the right place at the right time in a sense, but also making the decision early on that I wanted to give back to the NCI. And so I worked my way up, and I guess I did a good enough job that I was invited on to be on a program committee and a parent committee. And then, I was actually invited to be on the board of scientific counselors, which investigates all of the labs at the NCI. So I was getting a lot of understanding of how the NCI works, and I enjoyed it. And I was meeting all new people and learning how other people do things, which is important because just learning how I do it or how people at Hopkins does it was not going to be enough. I was also working my way up at AACR, and I was elected to lead the cancer immunology task force every year, a new person. So I was elected that year. And this was right before anti-CTLA-4 was going to be approved. So it was about 2010. And Harold Varmus, who is a wonderful person, but did not appreciate immunology at the time, was actually leading the NCI. And so I had this idea that, they weren't really putting a lot of funding into immunology at the NCI, very little into cancer immunology. So I had this idea that I would take my committee of people to meet with him. And so there was like 5 of us who show up in his office. He agrees to meet with us. And we're saying to him, do you realize what's happening? We're on the verge. And he's like, yeah. And, smart guy, wonderful person, but I don't think he was really getting it yet. And so I finally say to him, you realize that the ship is going to sail and the NCI won't be able to take any credit. So the next thing I know, I guess he put my name on a list. I guess he could have hated me. I guess, it's like, keep your friends close, keep your enemies closer, maybe, I don't know. But all of a sudden I'm on a list for, they submit lists to who should be appointed to the NCAB, the National Cancer Advisory Board. And the next thing I know, Obama's administration is calling me and saying, would you like to be on this? And they're evaluating me. And so I didn't get to meet him at that time, but I spoke with the people there. And I'm on the NCAB. And then what ends up happening is Varmus decides to leave, anti-CTLA-4 is approved, PD-1 is on its way. He leaves, and Doug Lowy, who, again, he's been one of my mentors as well nationally. He's just such a wonderful individual, virologist, developed the HPV vaccine. And so, he takes over interim, and he gives at the next meeting, he gives his kind of, where we are and what our priorities are. 45 minutes. And I remember because Chi Dang was on the board of scientific counselors and he was board of scientific advisors, and he was sitting next to me at the time. And, he's 45 minutes. Immunotherapy not mentioned, immunology not mentioned, genetics, all this other stuff. And I look at Chi and he says, yeah. So I raised my hand. I said, Doug, I said, this is wonderful, but the big elephant in the room is cancer immunology, and you haven't mentioned it. And poor Doug, he looked at me and and this is a open meeting, right? These are not closed meetings. So anyone could, take part. So, lots of people in the room. And he just looks at me and he says, "You're right." So anyway, needless to say, so then this opportunity for the Blue Ribbon Panel comes along, because now it's 2015, I think, and the State of the Union, and President Obama says to Biden, "You're going to lead the moonshot." So, this is when all of a sudden I get to be appointed. Again, I was on the list, I guess. I hadn't really met President Obama or Vice President Biden, but myself, Tyler Jacks, and Dina Singer led this committee, and we came up with a list of experts that spanned all different, expertise, and included diverse individuals. And we really spent a lot of time thinking hard about who to invite. And it was from like February until September that we were just lots of meetings, in-person calls, and I had to be a part of all of them, organizing and try to focusing people and pushing them in the right directions. And, Dina, Tyler, and I would meet, and then we met interimly with Vice President Biden. It was the first time I met him. Then I got asked, because now he knew who I was, so I got asked to join ACR in meeting with him. I wasn't president-elect at the time, but I was on the board of ACR. And so it was just a few of us meeting with him. And of course, he knew who I was at this point. And don't laugh, but Katie Couric calls me and says, because Stand Up to Cancer had taken off, and she calls me and says, "Can you get me in to see Vice President Biden? Because I want to talk to him about Stand Up to Cancer being involved in the moonshot." So Katie, Bill, and I go and meet with him in his office at the White House. And this is when he was being asked to run for president. And he starts telling us about this. And then he says, and he had tears in his eyes. He says, "I can't even think about that. I have to think about curing cancer. I promised my son." I mean, it was so genuine. It was really, really, it really hit me hard. And I had lost my husband to ALS at that point. And so, yes, my husband didn't die of cancer, but to be honest with you, I think he died of something just as bad, if not worse. Until he got ALS, I thought pancreatic cancer and GBMs were the worst cancers I've ever seen, worst diseases to get. And then he got ALS, and I realized that it was worse in one respect. It was worse because there was no hope. And I never understood the word hope before as well as I do now. Patients always want hope. And I felt I was giving them hope by giving them innovative new therapies that had the potential. But, new therapies, you had to still say to the patient, "It's unlikely it's going to benefit you," which was frustrating, but it was the truth. And I realized what hope really means. Even though they knew that it would unlikely benefit them, there was the hope. My husband had no hope. And Vice President Biden had no hope for his son, but he had hope for other people, and he really cared about that. And so that was kind of a connection we both made with each other. And then, we presented to him again the priorities, and I had to testify at Congress. And then also President Obama appointed me as the chair, and I think I'm the first woman chair of the National Cancer Advisory Board, which was also exciting. And I thought I was coming off of that when I accepted, to run for ACR president, but with the transition, so it never occurred to the new administration to switch out. So they were keeping me on for a while on the NCAB until, it got closer to the time when there would be change. So that was interesting. So I ended up staying on it for quite a while, which was actually interesting because I was still on it in chair when the pandemic happened. And so I helped to work with the NCI because there's a lot that had to be done for cancer patients as well. So that's my story. I did get asked to consider NCI director early on, to consider NIH director, and then to consider NCI director again. And the answer is I love working with all of them. I love the people at the NCI, but I love my job. I love mentoring the people I'm mentoring. I love research. We're at a critical time in, pancreatic cancer research. I want to be a part of it.
I mean, listening to hear that last reply and like all of the different ways that you have impacted the world of cancer research and care and advocacy, this last question seems a little bit unreasonable because you have had such an impact, and it seems like you're being very intentional about the path that you are taking. But if you had to do it all over again and you couldn't be a physician or a scientist, what other realm do you think could have kept you this passionate and this dedicated for a career?
It's an interesting question. I thought about that a lot. And I would tell you, after having kids, to be a teacher. Now, not kind of a teacher just teaching math, but developing new programs, really thinking about how do we teach children, as they mature, based on what we know about child development, based on what we know about new technologies. I would love to be involved in developing programs that would help young people develop, to their best of their abilities. I think that would probably been as satisfying and, to me, as important as what I do.
I love that there's still a scientific approach in there. All right. Well, Liz, thank you so much for taking this last hour to chat with me. It has been really, really wonderful to hear about the path that you took and the original thoughts and the intentionality that you've had. So thank you so much for all that you continue to do for the cancer community.